Welcome back, students. Today we are going to discuss part 13 of Unit 2, that is Cell as a Unit of Life. But before we start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of original creator and owner of these images. If you are the owner of any of the images used in this particular PPT, have any concern, any issue, kindly contact us at sathee@iitk.ac.in and we shall remove the images. In today's video lecture, we are going to discuss about summary of cellular respiration and oxidative phosphorylation. First of all, let's discuss the term cellular respiration. So, in previous two lectures, we have already discussed this topic in a very detailed way. But once again, just brush up the definition of cellular respiration. So, it is a process of using oxygen in the mitochondria to chemically break down organic molecule like glucose, right, to release the energy which is stored in the bonds of glucose. So this is cellular respiration. What is the definition? Cellular respiration is a process of using oxygen in the mitochondria to chemically break down organic molecule such as glucose. This release the energy stored in the bonds of glucose. This is known as cellular respiration. Okay, and during this process, molecule of water and carbon dioxide are released as a waste product. Okay, and during this process, in various steps, some amount of energy also released in the form of ATP. So some molecules of ATP has been released during this cellular respiration, which further cell utilize to function their various metabolic activity. Right. So this is cellular respiration. So what is the reaction over here? This is a glucose molecule C6H12O6. When it combined with oxygen, okay, or we can say when it utilize oxygen, then it will convert into water, carbon dioxide and energy. Okay, so this water and carbon dioxide both released as a waste product, and so much huge amount of ATP has been generated during cellular respiration. Okay, clear now. So what is the first step cellular respiration? First step is breakdown of glucose molecule, right? And this glucose comes from where? It comes from a carbohydrate, or we can say sucrose, right? Then this sucrose then broken down into its monosaccharide forms like glucose and fructose, and then further this glucose enter into the glycolysis pathway, and this particular pathway, this cycle occur in the cytoplasm of the cell. Okay, and then this glucose further convert into glucose 6-phosphate, then fructose 6-phosphate, and so on. This is a 10-step reaction. And during each step, during initial stages, this glucose actually utilize ATP molecule, as you can see over here. It utilize one ATP molecule over here, another ATP molecule over here. Why? To convert this glucose into glucose 6-phosphate and glucose 1,6-biphosphate. For this purpose, initially glucose utilized 2 ATP molecule, but in the last form or last steps of this glycolysis pathway, 4 molecules of ATP molecule has been generated, as you can see over here. This one and this one. 4 molecules, because this one molecule of 6 carbon, glucose is a 6 carbon molecule, further convert into 2 molecules of 3 carbon atom. That is glyceraldehyde 3-phosphate, and DHAP, dihydroxyacetone phosphate. Okay, and this reaction shows only one molecule reaction. So total 4. So what is the net gain? Net gain is equal to 4 molecule, 4 ATP has been synthesized during the process and 2 has been used up during initial process. So what is the net gain? Net gain is 2 ATP molecule. Okay, 2 ATP molecule, which one can utilize it for any metabolic activity or any purpose. And apart from this ATP molecule, 2 molecules of NADH also synthesized during glycolysis. So 2 molecule of NADH. Okay, but this NADH is not ready to give up its energy over here. It further go into the next cycle or next level, means within mitochondria, within matrix of mitochondria, and then oxidative phosphorylation. And during that process, this ATP, utilize it, that mitochondria utilize energy present in this ATP in the form of ATP, NADH to ATP. Okay, so here what is the main content? Initially, during first step of cellular respiration, that is glycolysis, this glycolysis occur in the cytoplasm of the cell. First important point. Then secondly, this sucrose first converted, sucrose is a disaccharide, convert into its monosaccharide form, means glucose and fructose, and during whole process of glycolysis, initial steps ATP has been utilized up, 2 molecules of ATP has been utilized up, but on the last step 4 molecule has been generated, 4 molecule of ATP has been generated. So net gain of ATP occur during glycolysis is 2 molecule of ATP. Okay, and apart from this ATP, 2 molecule of NADH also has been generated during this process. Okay, now, so 2 molecules of pyruvate. This is the last product of glycolysis. Okay, 2 molecules of pyruvate. What is the fate of pyruvate? It may convert into alcohol. It may convert into lactic acid, or it may convert into carbon dioxide and H2O. Means all the bonds which are present within the carbon-carbon bond of this pyruvate, all those bonds are having energy in itself. So all 6 bonds has been broken down during oxidative complete, or I can write down here over here, complete oxidation. Complete oxidation during aerobic respiration. So if oxygen is not present, means no oxygen is there, oxygen is not present, means in anaerobic condition, pyruvate can convert itself into alcohol, and a small amount of ATP has been generated during this process, only 2 ATP, that is already generated during glycolysis. Similarly, it may convert into lactic acid, and a small amount of ATP has been generated, which is already generated in the glycolysis, because, or in some kind of prokaryotic cell in which mitochondria is not present, right? So it may be utilized in this alcohol fermentation pathway, it may be utilized in lactic acid fermentation pathway, this pyruvate, or complete oxidation occur during the presence of oxygen or in aerobic conditions, right? So plus oxygen or aerobic conditions or aerobic organism, right? So now if mitochondria is there, if aerobic organism is there, if mitochondria is there, then this pyruvate, two molecules of pyruvate, then convert into acetyl coenzyme A, right? And then this acetyl coenzyme A, which is a 2-carbon molecule, then enter into this citric acid cycle, which is a main cycle performed in the matrix of mitochondria, right? And this acetyl coenzyme A combined with oxaloacetic acid and convert itself into a 6-carbon molecule, that is citric acid cycle. And that's why it is written over here citric acid cycle, because the first component, first type of molecule generated in this TCA cycle is citric acid cycle. And this TCA citric acid is having 3 carboxylic group in it. That's why tricarboxylic acid cycle, okay, or Krebs cycle, who first described this cycle. So anyways, we have already discussed this TCA cycle in our previous lecture. So after this citric acid cycle, there are so many other component has been formed, and during the formation of one to another component at 3 different level, NADH has been generated, first, second, and 3. Okay, 3 level. And so can I write down over here, during the conversion of pyruvate to acetyl coenzyme A, 1 NADH has been generated over here? Yes. So, and this particular reaction, the conversion of pyruvate to acetyl coenzyme A, I can write down over here link reaction. This is a link reaction because this reaction connects citric acid cycle to glycolysis. Okay. So, 1 NADH during link reaction and 3 NADH during citric acid cycle. So what is the total number of NADH in generated inside the mitochondria? This is the number 4. 4 NADH. Am I right? Yes. Okay. And FADH, 1 molecule of FADH. So 1 molecule of FADH also generated during citric acid cycle, right? And 1 molecule of this GTP or ATP. So I can write down over here ATP. So this is the calculation. This is the calculation for 1 molecule of pyruvate. While from 1 glucose, 2 molecule of pyruvate has been generated. So if somebody asks how many NADH or FADH has been generated by utilizing 1 molecule of glucose, then we can write down into 2, means 8 NADH into 2 molecule of FADH, NADH, okay, and 2 molecule of ATP. Correct? Okay, so this is the total calculation of NADH, FADH and ATP, which is generated during citric acid cycle and link reaction, both, or within, we can say, mitochondria. Okay, in glycolysis, 2 molecules of ATP and 2 molecules of NADH has been generated, which just now we have discussed. Now, next. So as you can see this is a glycolysis process. This is a glycolysis process where glucose gets converted into pyruvate, 2 molecules of pyruvate, and this is the 1 molecule, right? And during this conversion, some amount of ATP has been generated. Just now we have discussed how many? 2 ATP molecule. So this 2 ATP molecule is known as, generation of this ATP molecule is known as substrate level phosphorylation. Directly they have generated this much amount of ATP, although during this process some amount of NADH also generated, but that is not ready to give up the energy. as a ATP, right? That NADH, as you can see, this is NADH. This NADH also generated during this pathway only, but this actually carries their electron and go inside the mitochondria, and there those electrons utilized by the oxidative electron transport chain. Okay, and during oxidative phosphorylation, by the utilization of those electrons, ATP has been generated. Okay, so on the substrate level phosphorylation in the cytoplasm during glycolysis, only 2 ATP molecule has been generated. So can I write down this? 2 net ATP from substrate level phosphorylation has been generated during glycolysis? Yes. Right, and 2 NADH also generated, which will yield 6 ATP molecule during oxidative phosphorylation, as 1 molecule of NADH is equal to 3 molecule of ATP. So 2 NADH molecule will give us 6 molecule of ATP. But what time? During oxidative phosphorylation only. Clear? And during link reaction, just now we have seen link reaction. During link reaction, where pyruvate get converted into acetyl coenzyme A, that time also 2 molecule of NADH also generated, which further gives us 6 molecule of ATP by oxidative phosphorylation. Clear? So during this cycle, pyruvate to pyruvate oxidation, where in this citric acid cycle, acetyl coenzyme A only type of molecule which entered during this citric acid cycle, right? And during citric acid cycle, directly 2 molecule of GTP or ATP has been formed. Okay, so that 2 ATP comes under substrate level phosphorylation, 2 ATP molecule or 2 GTP molecule. While some other molecules has been formed during citric acid cycle as NADH or FADH2. So 6 molecule of NADH and 2 molecule of FADH2 formed. And with the help of this 6 NADH, we will get 18 molecule of ATP by oxidative phosphorylation, and with the help of 2 FADH2, we will get 4 molecule of ATP by oxidative phosphorylation. As I told you, 1 molecule of NADH is equal to 3 molecule of ATP. Similarly, 1 molecule of FADH2 is equal to 2 molecule of ATP. And that's why 1 molecule gives us 2 molecule. So 2 molecule gives us 4 ATP molecule. Clear? So what is the total number of ATP generated per glucose molecule in the prokaryote? This is 38 ATP. Right? Fourth, from the substrate level phosphorylation, just now we have discussed 2 from this glycolysis pathway, 2 from the citric acid pathway. Rest NADH and FADH2 gives their energy as a ATP molecule or convert ADP into ATP molecule during oxidative phosphorylation. Means 34 molecules from oxidative phosphorylation. This is very important point. Out of 38, 34 from oxidative phosphorylation. Is it clear? Now, okay, in this tabular form, once again, we have discussed the process. What is this stage? Glycolysis. Where it occurs? Cytoplasm. Am I right? What kind of molecule enter in this particular cycle? Glucose. Right? And what is the outcome of this cycle? What come out? 2 molecule of ATP. Correct. 2 molecule of NADH, correct, and 2 molecule of pyruvate, correct. Okay, so this is 2 molecule, okay, 2 molecule. 2 NADH means 2 into 3. 3 molecule of ATP just now we have discussed. So 6, right? So total amount of ATP 8. If we count total amount of ATP. Then in Krebs cycle, what is the second stage? Krebs cycle, where it occur? In the matrix of mitochondria. What kind of molecule enter in this pathway or matrix? This is pyruvate, right? So with pyruvate, 2 molecules of ATP. Yes, correct. 8 molecules of NADH, including that link reaction, right? Conversion of pyruvate to acetyl coenzyme may occur, first step, and 3 molecules during Krebs cycle, right? So with 1 molecule, and this is for 2 molecule. So 2 ATP, 8 NADH, and 2 FADH2. So can I write down 2 over here and 8 into 3, 24 ATP, 2 into 2, because 1 FADH2 is equal to 2 molecule of ATP. So 4. Can I write down over here? So what is the total number? 30 ATP. So during Krebs cycle, we can say, or in mitochondria, we can say, both the terms are clear, right? 30 ATP, and out of glycolysis, not directly to only 2 ATP we can utilize it. Just now we have discussed substrate level phosphorylation. Only 2 ATP direct we can utilize over here, or we can get, and we can get 2 ATP direct, 2 ATP from Krebs cycle, substrate level phosphorylation. Remaining other molecule just carry their electrons in a very vigorous way towards oxidative phosphorylation, F0, F1 electron transport chain, where so many carriers are there. From one carrier to other carrier, very quickly electron move from one carrier to another carrier, and during this process, they just reach to F0, F1 particle, where they convert. Because of that proton gradient, ADP and inorganic phosphate combine together and produce remaining ATP molecules. So what is the total number of ATP molecule generated during cellular respiration? Is this 30 plus 8 is equal to 38 ATP molecule. Is this clear, guys? Is this clear? Okay. So I have already mentioned this line. Already we have discussed this about ATP and NADH relation previously also. So this is the whole chart. Okay. So I think there is no confusion now. Okay. 2 direct ATP we can get from this glycolysis pathway, and 2 direct ATP we can get from a Krebs cycle, remaining as a NADH or FADH2, which further convert its energy into ATP during oxidative phosphorylation. Okay, so this is all about the cellular respiration. Now, what is oxidative phosphorylation? So this oxidative phosphorylation, which we have discussed so many times till now, this oxidative phosphorylation is an aerobic process of energy harnessing the production of ATP in mitochondria only by enzymatic phosphorylation of ADP coupled to electron transport chain. Okay, and this electron transport chain is a series of mitochondrial enzyme as a proton carrier molecule like cytochrome B, C1, cytochrome A, A3, cytochrome C. All those carrier molecule are there. We have discussed previously, right, in last lecture. So this ETC or electron transport chain is a series of mitochondrial enzyme present in the mitochondrial membrane, inner membrane, right? And high energy electron are shuttled down the chain via NADH and FADH2. These are known as high energy energy bonds these contains. Okay. Uh, so as high energy electron are shuttled down the chain via NADH and FADH2 to oxygen molecule, that time these electron molecule will produce ATP and water. Water is the final acceptor of electrons, and protons they just utilize the remaining proton which is present in the matrix part, right? So this is all about the cellular respiration. Now let's compare fermentation with aerobic respiration. As you know, this fermentation occur in the anaerobic type of cell like yeast cell, or uh, in that environment where oxygen is not present. So during fermentation, only partial oxidation of that particular glucose occur, or we can say partial breakdown of glucose. Whereas in aerobic respiration, complete degradation of glucose occur, means complete oxidation, or a complete cellular respiration, or complete oxidation is the right term, where it is means glucose completely degraded itself into carbon dioxide, water, and high amount of energy in the form of ATP has been released. We have just discussed this in my first slide. Glucose plus oxygen is equal to carbon dioxide, H2O, plus energy as a ATP. This is the formula. C6H12O6 plus 6O2 is equal to 6CO2 plus, okay, C6H12O6, 6O2, 6CO2, 6H2O, and 38 ATP during complete oxidation. This is the formula right now. Right, we have discussed in the during first or second slide. So fermentation is a partial breakdown of glucose, while in aerobic respiration, complete degradation of glucose occur in the carbon dioxide and water. Secondly, in fermentation, there is a net gain of only 2 molecules of ATP occur, because only that process occur in the cytoplasm only. Okay, so only 2 molecule of ATP formation occur during fermentation. Okay, so for each so one glucose molecule, from one glucose to 2 molecule of pyruvate plus some amount of ATP, means 2 ATP. This pyruvate further convert into alcohol, or it may convert into lactic acid. That's why there are 6 bonds in glucose molecule, only one bond is broken down. So very less energy, means only 2 molecule of ATP has been generated. Okay, and then this glucose degraded into pyruvic acid, whereas many more molecules of ATP has been generated under aerobic condition. Why? Because all the bonds which are present in glucose molecule has been broken down. That's why we can use the term complete oxidation during aerobic conditions. That is the third part where it will convert into carbon dioxide, H2O plus ATP. Huge amount of ATP, or I can write down 38 ATP. So this is the difference between fermentation and aerobic respiration. Clear? Now, as you know, plants don't have some special organ or some special system for breathing or for gaseous exchange as humans or animals have. We have nostrils, we have lungs. Okay, so whole respiratory system is there, but plants do not have these kind of system. They only have stomata which are present on the leaves, some lenticels, which are present in the, if this is a stem, if this is a kind of stem. So these kind of openings are present on the outer layer of stem, and these opening is known as lenticels, which actually allow gaseous exchange. And further, they with the help of this diffusion, they can pass gas from one cell to another cell. Okay, so almost all living cells in the plant cell have their surface exposed to air. Um, but in roots they have loosely arranged parenchyma, so very easily they can pass one gas from one cell to another cell. Stomata present in the leaves, lenticels present on the stem and other hard part, while in the roots they have loosely arranged parenchyma. Okay, so this is how they can doing their we can say gaseous exchange process. Okay, and the breaking of carbon-carbon bond of complex organic molecule by oxidation cell, leading to release of lots of energy, which is called cellular respiration. Just now we have discussed about it. If all the bonds have been broken down during cellular respiration, during aerobic respiration, or complete oxidation, we can say, only that time lot of amount of energy has been released. And glucose is the favorable substance for respiration, right? Although if inside a body, inside a cell, glucose is not present, that time cell utilize fat or protein to generate energy. Okay, these fat or protein also broken down with the same pathway. Some initial steps has been different although, but these type of molecules also broken down to yield energy. Okay, the initial stage of cellular respiration takes place in the cytoplasm, means glycolysis. Though all the steps present in that glycolysis path occur in cytoplasm only. Okay, these are some other important points related to this particular topic. Now, let's briefly discuss summary about this whole topic, cellular respiration. So, the energy in these molecule means all the molecules which has been generated from glucose to other three carbon molecule or two carbon molecule, like this. So in summary they are discussing about those molecules. Okay, so the energy in these molecule as well as that in the NADH synthesized during glycolysis are used to synthesize ATP. Yes, very true. And this is accomplished through a system of electron carriers which are present in electron transport system. And these electron carriers are located where? On the inner membrane of mitochondria. All the carrier molecules are located in the inner membrane of mitochondria. True? Yes. The electrons further move through the system, this electron transport system, release enough energy that are trapped to synthesize ATP, and this movement of electrons is known as oxidative phosphorylation, or synthesis of ATP is also known as oxidative phosphorylation. Okay, and during this process, oxygen is the ultimate acceptor. Oxygen is the ultimate acceptor of electron. Clear? And it get reduced to water. After accepting electron, this oxygen get reduced as a water, or we can use the term metabolic water for the same. Okay, the respiratory pathway, this whole pathway is an amphibilic pathway, as it involves both anabolic and catabolism. And this is very, very important point. Means if glucose is there, after so many steps, it will get convert into, pyruvate, and then pyruvate into acetyl coenzyme A, and then this enter into Krebs cycle, and further all the steps has been started. But this pyruvate may enter into another pathway. Means this is the catabolic pathway. Glucose, this 6 carbon molecule get convert into 3 carbon molecule, then 1 carbon molecule, and then finally carbon dioxide to water, 1 carbon molecule. But it can also enter into another pathway, which in which this pyruvate or oxaloacetic acid enter in that pathway and start to synthesize fatty acids or and amino acids, sorry, and so on. That's why we can say this respiratory pathway can be a catabolic pathway, or it's any intermediate product can be utilized or enter into another pathway, another biosynthetic pathway, where it can um, utilize for the synthesis of fatty acid or amino acid or other components. That's why it is written over here. The respiratory pathway is an amphibolic pathway, as it involves in both anabolism and catabolism. That's why amphibolic term has been used over here. Clear? Okay. So this is all about the cellular respiration. This particular chapter is very, very important. There are so many uh, question come in various examination related to this chapter. I hope all the doubts, all the small concepts, or whatever present, whatever syllabus you have in this particular chapter I have covered, and all the doubt has been cleared now. Still you have any query, any doubt, you can write down to me in comment box. We will discuss that doubt later on. Till then, take care. Bye bye. Keep learning. Namaskar.